All right, welcome back, everybody. We're going to start session 2. This section is on cellular molecular engineering. I am Lance Kam, chair for this session here. I want to thank the organizers for making this possible. And I have to admit, when we first put this together, I'm like, I have to follow a session with AI and looking at some of the other things coming up. This is going to be a hard task. I mean, we know cellular molecular engineering, this is one of our original pillars of the department and things. But as we got it together, I mean, I'm excited about this program. The collection of speakers we have really shows what the forefront of this field is. It's exciting, vibrant. We don't need a fancy name on this session. And so we'll get right to the speakers. We're going to start off with Harris Wang. Let me see, he's associate professor and interim chair of systems biology. We've been talking about centers and things, how we bring together engineering and medicine and many of the things that we have here. Certain people, like Harris, bring it together himself. And so we're getting the equivalent of a center here. So I'd like to just turn the floor over to Harris, please.
Great. Thank you, Lance. Can everyone hear me? This is okay. Okay. Yeah. So today I'm going to speak with to you about some of the work that we've been doing in our lab in thinking about how we can engineer and edit the microbiome and potential kind of therapeutic opportunities for that. Just quickly, these are my disclosures and funding sources. As many of you know, kind of the gastrointestinal tract is actually a really interesting organ in the body. It's actually what I think about is kind of this the body's portal to the molecular universe. Every single molecule is essentially that goes through your body goes through the gut, right? And so at this interface sits not just kind of immune cells that are quite abundant and really important for kind of protecting the body from the other resident members, which are essentially kind of these microbes that there's potentially trillions of microbes that sit at this interface. And so it's such a really interesting organ because it's at this nexus where kind of the outside world meets the inside world of the body when the body needs to be able to interact with it, be able to extract molecules and nutrients for a variety of functions. And so, and it's all very clear that the microbiome itself has a lot of implications with regard to human health and also in disease. And so there's been a number of kind of clinical trials that have been initiated, and there's specific commercialization of these microbiome-based therapeutics, including FDA-approved microbiome fecal transplantation, where you could essentially take a healthy microbiota and be able to transplant it into individuals, for example, that have received or are infected with a pathogen that causes very severe gastrointestinal distress and potentially death. And so, as many of you also know that kind of the microbiome is just part of a larger repertoire of genetic material that exists within our body. A lot of current effort has actually been focused on trying to edit the human genome, for example, in gene therapies. And there's a number of advances associated with CRISPR technologies that have now been approved. And so, but what's interesting is that the gut itself and the microbiome holds this vast genetic reservoir of potential metabolic capabilities that are actually vastly outnumber both in numbers of cells to human cells, but also in terms of genetic capabilities. And so we think kind of this central bioreactor organ is really important facet that we know is linked to disease. But the major challenge here is that this organ is composed of hundreds, if not thousands, of unique different strains that collectively make up, what people call this kind of meta organ. But it's actually very challenging to be able to actually manipulate any individual components of this. And so kind of the thesis of the lab, or at least a part of the research that we're doing, is to try to develop technology to be able to manipulate this what we call a meta organ. And so kind of inspired by this idea that certain organs we can't actually extract and be able to do things outside of the body, we need to actually be able to go in and precisely edit and modify that. Essentially gene therapies, we kind of thought maybe we could actually develop an approach very similar to gene therapy for microbiomes. We call kind of this kind of surgical precision gene therapy for the microbiome, where we can deliver very precisely specific genetic functions via bacterial conjugation system that can then transfer these genetic elements, and then those could get integrated into very specific members of the microbiota. And this requires essentially technologies that are developed in my laboratory as well as by our colleague Sam Sternberg's group at the biochemistry department at the medical center, where he developed this very interesting, or he discovered and then further developed this very interesting CRISPR-Cas technology, where it combines the RNA-targeted capabilities of Cas systems with integration transposon system that allows very precise programmable integration of very large genetic payloads arbitrarily into any sequence of interest here. And so with this, we think we have now this capability to do what we call surgical precision gene therapy. Just to kind of show you a few data points for this, within the microbiome, there's potentially gigabases of actual genetic material, just like there's gigabases of the human genome. And so we can precisely now land different payloads into any number of microbes within that community at very high fidelity, 99.8% of specific microbe. And then we can actually sorry for the pause here. I think the screen sharing isn't working. We can show that these genetic these strains could be individually targeted, including ones that actually we are not able to grow in the laboratory. So they can be grown in the gut. We can't actually bring it out of the gut into a laboratory setting, but we can directly do in situ editing, including this segmentus filamentous bacteria here, which is known to have kind of major impacts on kind of gut immunity and entrainment of immune systems. I'm going to start telling gut jokes. Okay, we're good to go. All right. Okay, so one of the other things we showed is that not only can you target these specific strains within the gut, we can also potentially control it with some external sources. For example, inulin is a dietary metabolite that sometimes is consumed. And we essentially took a genetic payload that allows the bacteria to be able to now metabolize inulin. And we put that precisely into a specific strain that exists within the gut. And now we can effectively modulate the colonization or the level of activity of this particular strain by just applying externally inulin. And this is what I'm showing on the figure here, where when we give inulin supplementation, we see the expansion of this particularly edited bacteria. And when we remove that inulin, that genetic functionality is now removed. And this is really important for us in terms of building some safety controls here compared to kind of the control on the bottom here, where the genetic elements is essentially removed. And this allows us to develop kind of genetic gene drive technologies. And so this gene drive technologies allows us to put in arbitrary functions of interest and then drive that completely and transform the entire microbiome with that genetic function of interest. And it can do kind of primary and secondary and tertiary transfers of this functionality. And just to kind of give you a couple of different applications of this technology, as maybe a few people saw recently, there was a big E. coli outbreak at McDonald's, and so a lot of people got sick, and it's caused by this Shiga toxin or bacteria that produce Shiga toxin, Stec bacteria, which actually leads to a lot of very kind of nasty kind of syndromes, some of which are quite fatal, especially in kind of developing worlds. This really has no actual kind of treatment. Essentially, it's your doctor says get some hydration and try to just palliative care. Importantly, you can't use antibiotics because if you try to kill the bug, then you actually release a lot more toxin, and that's actually even worse, right? And so the idea here is that instead, if you get colonized with this pathogen, we're going to try to go in and in a targeted manner destroy this particular Shiga toxin gene using this gene delivery and editing technology. And so within this pathogen, there's a few different genes, and we identified the specific subsets of this that's actually quite potent, and that's actually shown on the bottom right there, where if you knock out, for example, the XTX2B gene, you get significant decrease in the overall Shiga toxin levels within the gut. And that provides very great protection. Normally, in a mouse model, these mice essentially die within 5 days of being infected with this. Strains that don't have this toxin essentially are completely protected. The other thing about this is that we not only can kind of remove the functionality by knocking in this genetic cassette, we can also have the genetic cassette itself have some therapeutic capabilities. In this case, we have it essentially prevent attachment of the Shiga toxin E. coli by inhibiting its by expressing an antibody that prevents its surface attachment along the gut. And long story short here, we were able to show that this combination of technology significantly provides protection from this otherwise very lethal dose of the Shiga toxin. In fact, we have a number of animals that survive over a long period of time, and actually the survived individuals actually are now protected from subsequent infection. Now, just to highlight here that normally in humans the lethality is not 100% within 5 days. So in an animal model here, this is very drastic, kind of very stringent phenotype here, and any level of protection would indicate that there's likelihood that this would also work in humans. Just very quickly, the second application here is around GLP-1. As many of you know, this is a very hot drug in terms of for weight loss and for and so in the context of this, we actually also consider GLP-1 as a candidate to improve barrier function. And it was actually observed that there's a skin bacteria that normally lives on the skin that helps wound healing on the skin, but it has this delta toxin that otherwise is able to stimulate production of GLP-1. And we essentially took that delta toxin peptide and we put that into an E. coli Nissl, which is a probiotic bacteria. And then we showed that essentially this actually prevents colitis associated lethality as well as just negative effects of gut inflammation. And so when we give bacteria that contains this delta toxin, and we give it some gastrointestinal distress, these animals have kind of improved weight loss. They survive better. There's an increased level of plasma GLP-1 levels, and then there's generally decreased inflammation markers all around. And then overall, we showed that when we specifically knock out the GLP-1 receptor with an antagonist, this effect goes away. And so this indicates that the therapeutic kind of route is indeed through this GLP-1 kind of track here. There's lots of new opportunity for editing the gut microbiome. I mentioned a couple of these things, but we think we're very excited about kind of the utilization of this technology for a variety of different applications here. And so with that, I think I'm out of time here. Just want to thank my funding sources, the folks in my lab, and for you all for your attention. Yeah, great. Thank you.
Thank you very rapidly, and then through things like this is beginning to profess. So be a professor, yes. So we turn the floor over to her.
Okay, I think I'm good. So in today's talk, I would like to give an overview of my lab's research on engineering more effective biomolecular immunotherapies. So immune system is a complex network of cells and organs spread throughout the body. Simply put, its main function is to recognize threats, which can be pathogens or abnormal cells like cancer cells, and activate an appropriate immune response to eliminate these threats and then resolve the response. And ideally, it will also form a memory that can get reactivated quickly upon re-exposure. So in immunoengineering or immunotherapy, we aim to modulate and reprogram these immune responses to treat diseases like cancer, autoimmune disorders, and infectious diseases. It's not a new concept, so one of the oldest form of immunotherapy is vaccination, which has shown great success in curing or even eradicating different infectious diseases. Another form is cancer immunotherapy, which has made great progress in recent decades and can provide long-lasting survival benefits. But these effects are not consistent. For example, in case of seasonal flu, the vaccine effectiveness fluctuates each year. It's often less than 50%. Or with cancer immunotherapy, only a small fraction of patients show durable responses, and there's an obvious need to increase the percentage of these responders. So to address these challenges, my lab's research centers on developing biomolecular immunotherapies that can act at the right time, right place, and in the right way to maximize their synergy with the endogenous immune response mechanisms. To achieve this, we take a multidisciplinary approach, integrating synthetic biology, biomolecular and protein engineering, and biomaterial science. So, and today I'll share with you guys our efforts on how we leverage synthetic biology to control the timing of immunotherapies. So, but first, let me tell you about our biomolecular platform. So among biological therapeutics, we primarily focus on RNA, followed by protein. And these each have their own advantages and challenges. So protein therapeutics are generally more stable, they're easier to deliver, whereas RNAs are prone to degradation by nucleases and they need a specialized delivery system. RNAs can also trigger innate immune responses, but this can be a pros or a con depending on the context and the application. But what makes RNA our favorite platform is its ease of production. So it can be synthesized in vitro without needing any living cells, and it's scalable and cheaper, relatively speaking, and also faster to produce. So a great example is the COVID-19 vaccine. Once the virus genome was sequenced, according to the researchers behind it, they were able to make the vaccines in just a few days. So to be a little more specific, we work with a new modality of RNA called self-amplifying RNA, or saRNA, or replicon. So with conventional RNA, so it's injected into the cells and it's translated to payloads, which can be proteins, vaccine antigens, and cytokines. With saRNA or replicon, once inside the cells, it can also replicate itself. So it allows for a high level of payload expression from a small initial dose. This technology has been gaining a lot of traction in recent years. It has already found its way into clinical use. It has been approved for COVID-19 in Japan, and it's also in clinical trials for cancer treatment. So because of the self-amplifying nature of saRNA, so it allows for sustained gene expression. So in mice, we can detect gene expression from saRNA for over a month after a single injection, which is significantly longer than conventional RNA, where gene expression lasts for only a few days. So given this prolonged gene expression, we wanted to see if we can devise a strategy to control the kinetics, essentially the time and the level of gene expression from siRNA to further enhance its therapeutic efficacy. And this was motivated by numerous previous reports which showed that the sequence and also the pattern of exposure to immunotherapies can significantly impact the therapeutic outcomes, both for vaccines and also for cancer immunotherapy. Specifically for vaccines, it has been shown that with protein vaccines, if we inject the vaccine antigen or adjuvant in increasing doses over time, it can generate a stronger T cell and B cell responses compared to when you do constant dosing or bolus injections. But you can imagine that these repeated injections, they are not practical for mass vaccination. So we wanted to see if we can mimic these dose escalation pattern with our saRNA platform, but with a single injection. So to do this, we incorporated a synthetic biology-based small molecule responsive gene circuit in our saRNA design. So that the gene expression can be turned on or off or be adjusted by oral administration of small molecule drug. The gene circuit we selected was responsive to trimethoprim or TMP, which is an FDA-approved antibiotic. And this circuit is based on a destabilization domain or DD. When we fuse this DD to our protein of interest, the whole fusion is degraded by proteasomes. But binding of TMP makes this fusion stable and allows for its expression. So I'm not going to go into details. So we incorporated this circuit in our saRNA design to control both the saRNA amplification and protein translation. So when we have no TMP, the replicon cannot amplify itself, and also any translated protein is also rapidly degraded. And these turn the gene expression off. But with TMP, the circuit activates, turning gene expression on. So testing these circuits in mice, we showed that with using this circuit, we are able to achieve high levels of gene expression in the on or plus TMP state and minimal or non-leaky expression in minus TMP or off state. The circuit is reversible. We can have it on initially and then turn it off by removing TMP, or the other way around. We can start in the off state and then turn it on by introducing TMP. The gene expression level can be also fine-tuned by adjusting the TMP dose, and this control is maintained over time. So this circuit enabled us to achieve distinct gene expression patterns by adjusting the TMP dose. For example, we can have delayed expression where we keep the expression off for up to 2 weeks by withholding TMP and then starting the expression by adding TMP there. We can have escalating pattern by gradually increasing the TMP dose over time. And we can have more complex patterns like oscillating expression by alternating between high TMP and no TMP. So finally, we use this circuit for the expression of an HIV immunogen called EOD, just a model antigen. And we immunized mice, and we put mice on 3 different TMP regimens: constant low TMP, escalating TMP, and constant high TMP. And what was interesting was that when we looked at the germinal center responses in the lymph nodes of these mice, in terms of total B cell responses that we are getting, we didn't see any difference between the 3 different regimens. But our dose escalating regimen was able to induce a higher percentage of antigen-specific B cells. So basically, we are able to enhance the quality and the specificity of the immune responses that we are getting to vaccination using this strategy. So in my lab, building on this platform, we will incorporate synthetic biology approaches to engineer next generation saRNA based immunotherapies for both vaccine development and also for cancer immunotherapy. We are also interested to understand how the endogenous biological systems, for example, the innate immune responses like interferon responses, interact with and modify RNAs, especially saRNA, and harness these endogenous systems to make better RNA therapeutics. And finally, another key area of interest is to engineer slow-release vaccine formulations to better mimic the prolonged antigen exposure that happens in natural infections. So with conventional vaccines, unlike natural infections, the antigen sticks around for only a few days, so this can limit the efficacy of vaccines. So we are interested to develop RNA and also protein-based vaccine formulations that after injection, they can form a depot and release the antigen gradually and over an extended period of time. So that brings me to the end of my talk. I hope that gave a good picture of the research in my lab, and I'm looking forward to opportunities for collaboration and obviously excited to recruit the students to build our team. Thank you.
Okay, thank you for that. The next speaker in this is Ivan Vapi. He's from many departments, kind of embodying the interdisciplinary spirit of this session here. Affiliate appointment in BME, but also physiology and cellular biophysics, and way on the other side on dermatology. That's quite a collection. So we look forward to your talk.
Good afternoon, everyone. Thank you. I'm Ivan, as you heard. Can you guys hear me okay? It's working? We're going to talk today about regeneration. So I'm super excited about that. Our lab launched about 2 years ago. And as you heard, we are between 3 departments. Physiology, we do systems physiology work, cellular biophysics, and BME. Our model system is the skin, so we're going to get into dermatology context in a little bit as well. Okay, so bring a pointer. Okay, awesome. I'm just going to set up the pointer. Yeah. All right. So every multicellular organism has developed a mechanism or process to repair damage, right? Living in the natural world from plants. And very recently, actually, it was shown that dinosaurs had a very evolved way of doing this also in the skin. And so because of this phenomenon, multicellular organisms have developed really sophisticated ways to recreate tissue. And it goes from a spectrum, as you can imagine, from repair, which is sort of healing with some defects like scar formation, all the way to regeneration, which is something that we see also very commonplace actually in the natural world. And in people, because of living in the natural space, one of the leading causes of death in the U.S. is actually unintentional injuries. And it turns out that about 50%, over 50% now of all clinical visits actually have to do with wound caring and some procedure related to wounding, surgeries, birthing parents, soldiers returning from combat, and so many other conditions. This is actually Marcella Towson, this is just published by New York Times back in October. She was she became a victim of an explosion at her home, octane explosion, that burned her 2nd to 3rd degree. And what physicians did for Marcella was to actually apply placental grafts to the site that was damaged. And as you can probably tell, the face, there's a before picture that I don't include in here, but the face was severely destroyed. And this placental graft was able to restore Marcella's skin and tissue to virtually an indistinguishable physiology. But you can also notice the area of her body that was not treated, how defective that remains. And the reason I bring this up is because in regeneration science, we are really at a great limit of therapeutics for patients. And this category of treatment that Marcella received falls under something called replacement therapy. And replacement therapy is really what we mean by regeneration, actually, in the clinic. It just means we do not know how to regenerate complex tissue structures. We're just going to replace it with something that's already, that nature already formed. So if it's an external damage, like the one we just showed for Marcella, it's going to be a skin graft usually obtained from a healthy part of the patient. If it's an internal organ, the patient is going to receive a donor tissue. Typically, this is going to be a healthy human donor, but more commonly, you probably have heard on the news, transplantations from animal sources. If it's a larger structure, say a limb or something of that nature, prosthetics are commonplace, and if the patient qualifies, limb transplantation is also performed. So this is the current state of affairs. We do not regenerate complex tissue structures. And so my lab really was created to think about really outside of the box approaches to bring to bear gene therapy, gene editing, to regenerate very complex organ structures. And so that's what I'll be talking about today. Now, the way we can think about the therapeutic approach that currently exists is sort of a linear progression model or summation. So the example I always provide to students is, think of a scenario where you have two pieces, right? Two Lego pieces. So one piece here is blue. We add it to a second piece here in green, and the two of them form a new unit, right? This block. But this block can be summarized or can be described as the addition of these individual pieces. So it's a nice linear process. And this is actually a therapeutic model for regeneration now. So we have a patient with a defect or some kind of injury. That's block one. That's unit one. Then we add a second piece to the patient, which is the prosthetic, and that's unit B. And there are advantages to this, of course. It's very modular, it's easy to manufacture things at scale. The challenge with it is that it's not synergized within the organism. And so if the patient grows or shrinks or whatever have you, the blocks, in this case, will no longer be fit. So how does nature solve this problem, right? Because we all have limbs and they adjust to our size, et cetera. Well, that's the concept of emergence. It's a uniquely biological process where cells, billions of years ago, came up with a capacity to evolve complex physiological capacities or abilities by simply merging together and forming altogether distinct structures. So you have cells that can become organ systems, and this is happening all throughout our body continuously. So how can we engineer emergence, right? It's an elusive concept, and it requires just really sort of data sets that don't exist. So part of what I'll talk to you about today is our lab's attempt to create a data set of emergence and to build gene editing therapies or technologies to facilitate this. All right, so let's talk about emergence. So in the example of the Lego, you have two pieces, but this time, rather than these two pieces forming one block, they're going to form a sphere. So you go from red to green to a red sphere. So the structure, the product structure cannot be explained by an addition equation. That's part of why it's so elusive. But in nature, emergence is a very commonplace process. This is an example in salamanders. After an amputation takes place, there is dedifferentiation at the amputation plane, and cells at that site regrow to form the limb. They orchestrate this emergent phenomenon. Now, the field has tried to do this in the case of skin wounds. This is work by a couple of labs. One showcased that by trying to reprogram scar-forming cells inside of a wound site of a mouse, they were able to reduce the amount of scar tissue and actually increase the amount of epithelium. So that was one strategy. A couple of years later, we had work where scientists here, this is Dr. Longacre's work at Stanford, deleted a mechanical transduction gene and showcased that we could reduce scar formation and enable regeneration of hair follicles, complex skin structures. So we then have Michael Levin out of Boston, who showed that by having a cocktail of ion channel modulators, they could regenerate not only soft tissue, but also bone tissue in a frog amputation model. So these are all indications that a capacity for large-scale tissue reconstruction is possible, certainly in vertebrates. And can we take that to the next step in mammals and perhaps even people? So in our example, we have a patient here with a deep wound. Our lab sort of imagines, what if we understood every cell present at that injured site? What if we could characterize the genes that they express, the populations that emerge? And more importantly, what if we could actually synthetically orchestrate the process of emergence ourselves and take the tissue that's once damaged into the healing phase? And so as a consequence, we sort of focus on two different arms of our work, like I alluded to. The first is going to be atlasing the process of healing, so creating a map of emergence. And secondly, is going to create the technologies to facilitate it. So I began my talk I was introduced by noting that I'm in dermatology, and that's because I study this process in the skin. The skin, I think, is the excellent model system to investigate these questions because it is the first organ to have evolved. It contains everything from muscle to adipose tissue, immune cells, sensory neurons, and everything in between. And so the capacity to regenerate this tissue can give us a lot of insight about what can take place in other parts of the body. So First, I want to show you about some of the new gene editing systems that we developed to manipulate this process of emergence in the skin. This is CRISPR Spark, which is a system we recently created where we append a fluorescent tag onto an allosterically regulated Cas9. That just means that this is a Cas9 protein with a ligand binding domain fused onto it, so that only in the presence of the ligand can the Cas9 be functionally active. In this case, the function that we're going to study is its capacity to translocate to the nucleus to do any kind of editing. And so we've been able to demonstrate that by using CRISPR Spark, we can essentially control with very high titration the exact amount of protein that goes to the skin cells to change the genes. So once we have the technologies to temporally control genes, the question becomes, what genes should we control? And so our lab, also currently in preparation, has constructed what we call OA, which is an atlas of the entire wound healing sequence in mammals. It goes all the way from the uninjured state to the remodeling phase. And in OA, we capture every cell, every tissue that's part of the skin, and in temporal scale. So we know exactly what cells are recruited, what genes they're using to enable the reformation of the tissue. Now, we're using OA, we've created an atlas of tissue-specific promoters or cell type-specific promoters that we could use to actually molecularly dissect and control the fate of these cells. And so that's sort of where our lab currently is. We're now attempting, hopefully next time I come, I'll tell you about a publication coming out, of us attempting to actually recreate this process ourselves to see if we can actually heal the skin completely synthetically or artificially. Okay. Oh, yes. So my lab website is building sort of this huge database for the field. You go check it out, get to play with the skin, learn about the layers and the different genes that get expressed. And, of course, I'd like to thank the members of my lab. Thank you.
All right, thank you. For the last talk in this session, where is he? Professor Santi Carrera. His work really exemplifies this engineering in medicine kind of thing. We're going to hear about his work here, but I want to emphasize he's also involved in maybe the harder part of this, education as well. He's Assistant Director of Education and Training at HICCC. So you'll be working with him a lot. But here you go. Thank you.
Thanks, Lance. Hi, everyone. Thanks for being here. Is this the pointer right here? Okay. So it's a pleasure to be able to share my research with you today. So today we're going to be talking a bit more about biomaterials, and specifically how we can build biomaterials that have biological functionality relevant for reprogramming the immune system so we can deploy it to treat human disease. And so really the material system that I'm going to be talking to you about leverages fundamentally this principle of self-assembly, right? And so if you're familiar with nanomedicine, you might be familiar with this concept here of using molecular building blocks as a way to build these structured nanoscale objects that we call nanoparticles, which you can use for drug delivery applications. But what we'll focus on today is how we can actually think about nanoparticles as building blocks in their own right to construct larger macroscopic materials with unique functionality. Specifically today I'll talk about making hydrogels out of nanoparticles. And this is a class of material that is referred to as a supramolecular material. The bonds that form it are reversible and dynamic. That gives rise to some very useful properties, right? So in a technical capacity, we refer to them as shear thinning and self-healing. And that's just a way of saying that the material is injectable. This is really, really useful for local tissue engineering and drug delivery properties. And as I'll show you, this is also very beneficial for immune engineering. And so my lab, which was established here for about 2 years ago, I started at the same time as Yvonne, we think of these nano-engineered biomaterials almost as a thermostat for the immune system. But rather than controlling hot and cold, we're controlling immunogenic to tolerogenic outcomes. And so when I say immunogenic, I mean we're revving up the immune system. This is useful for treating conditions like cancer or potentially for developing therapeutics like vaccines. Or we can think about promoting tolerance or the tolerogenic side of the immune system, and that's helpful for combating autoimmune diseases. Again, we think about this in terms of how can we build a material that speaks to the immune system and convinces it to essentially re-regulate itself towards one of those two polar ends of its spectrum. And what I'll focus on today is how we can think about doing this with these nanoparticle-based materials, which is something that I actually became interested in my postdoc when I was at Stanford. And so I'll just preface some of the talk here with some of my work from that time, where I was working with a very inert polymeric nanoparticle hydrogel system. So this is a PEG-PLA nanoparticle. It's very safe, very biocompatible. You can mix this with a modified cellulose polymer, which is a type of polysaccharide. We introduce some hydrophobic chains onto that cellulose backbone. And when you mix those two components together, they interact and bind together almost as if there's a type of molecular Velcro. And that gives rise to these hydrogels that are really useful for delivering biological cargo, like antibodies or cytokines, which are often the way that we approach immunotherapy in the clinic. And I was really specifically interested in an immunostimulatory antibody known as a CD40 agonist, which had been used in the clinic for some time in clinical trials, but it keeps running into an issue of it being too toxic. And the reason for that is when you're doing something like cancer immunotherapy, you have a spatial problem. In the tumor, the immune system is not active enough, but in the rest of the body, the immune system is behaving relatively normally. But if you stimulate the entire body's immune system, you get these toxic side effects. And so what I wanted to see is if we could actually prevent some of those things from happening by using a biomaterial, in this case these nanoparticle hydrogels, to locally deliver that agonist to the tumor. And I wanted to compare that to the way that clinicians typically explore local drug delivery in trials, which was administering the antibody as a local bolus in a saline solution into the tumor environment. And what you're seeing here in this image are PET biodistribution studies. So this is where we can track a radio-labeled version of this antibody and see where it ends up in these mice that we're treating. And these mice are bearing a highly immunotherapy-resistant form of melanoma in their flanks. So I'll bring your attention to the left side. This is the local bolus. So this is what people would typically try to do a local therapy with this antibody without a material. And you can see within 24 hours, we're starting to see antibody accumulation in distant tissues, which is not ideal. This is how you start getting those toxic side effects. But when we look at our hydrogel, we're retaining that antibody very effectively in that tissue site. And as you'd expect, this leads to benefits in the safety profile. So in red, we have the local bolus administration of this antibody drug to a mouse, and it causes a rapid and acute weight loss, which can be quite severe. At the same dosage, if we administer that in a hydrogel, we mitigate that toxic weight loss side effect. And we also see benefits in the efficacy of the treatment. So in blue, with our hydrogel data, we're reducing the rate that the tumor is growing. And we also see a moderate but significant benefit in therapeutic overall survival when we administer this antibody this way. And one thing that's worth noting, and one of the things that I think is really exciting about thinking about drug delivery in this way, is that we've made this agonist antibody safer to use. So now you can explore different options that would be more aggressive, right? So introducing a combination therapy approach. So when we overlay immune checkpoint blockade onto this hydrogel, we now quadruple the rate of number of overall survivors in this highly immunotherapy-resistant form of cancer. And this is after a single round of therapy. And so this was all very exciting to me. I was really inspired by this because we were able to achieve these effects with a really simple system. It's just kind of a sponge for the antibody, and it sits there in the tissue and releases it over time. And I wanted to see what might be possible if we explored building these materials with more sophisticated nanoparticles. And so I brought to bear some of my expertise from my PhD and started looking at how we could introduce more complexity into this system if we had a liposome as a building block. And one of the things that's really nice about liposomes compared to those polymeric nanoparticles is that it's much easier to engineer their surface chemistry. And so what I started looking at was essentially different ways that we could encode interactions between our protein cargo and the liposomal crosslinks within these gels. And so here in this schematic, you can see kind of the three scenarios we explored: a situation where there's no interaction with our protein, this would release quickly; a weak interaction through electrostatic interactions, which would slow down release; and then our slowest release, which would be mediated by these strong receptor-ligand interactions. And when we look at this in vitro, and we confirm this also in vivo, we see this ability to control and finally regulate release properties over a wide dynamic range. And so this is around the time that I came here to Columbia to start my own independent group. These are some of the main research thrusts that we're exploring, but what I'm going to focus on for the remainder of the talk is a project in my group that's been exploring how we could make some of these materials using cell-derived nanomaterials. And so you may not know this, but you are a factory for nanoparticles. almost every cell in your body is producing nanoparticles to communicate with other cells in your body. And those nanoparticles are enriched with bioactive molecules like proteins, glycans, and even nucleic acids. These are often referred to as exosomes or cell-derived nanovesicles or extracellular vesicles. And for a long time, I've really been interested in trying to generate a hydrogel using this as a building block, because it should form a gel using a similar principle to what we pursued with our liposomal hydrogels. And you can imagine that if your nanoparticle building block has this intrinsic functionality, then you can introduce that functionality into your hydrogel and explore how that could be useful in very local drug delivery approaches or different tissue engineering approaches. And so I thought about this problem for a really long time, but really no headway was made until I got some really great trainees who started working on this project. And this was a very cool collaboration. One thing I'll point out is that we had a visiting scholar from Italy who actually heard about my lab and this project when I gave this talk 2 years ago over Zoom. And so she reached out to me to coordinate a stint here to do research, where she collaborated with a PhD student in my lab, Artemis, to drive this work forward. And so one of the things that we needed to overcome was really kind of a technical challenge. These exosomes or extracellular vesicles are very difficult to isolate and to concentrate and get at very high levels. Now, this is an issue in most kind of ways that we need to use them. People want to use them for drug delivery vehicles and things like that. But for our lab, this was especially problematic because when we're making a hydrogel from these materials, anywhere from 2 to 10 weight% of our hydrogel is actually the nanoparticle crosslinker. And so this seemed like a really insurmountable challenge for some time. And Kate actually had a really innovative way to try and solve this issue. So it turns out cows also produce nanoparticles all the time, and those nanoparticles end up in their milk. Kate had been working in her PhD with exosomes and was running into similar scale issues, and so she went to her local Parmesan factory, again, Italy, and they produce whey as a byproduct there, and they just gave it to her, and she was able to isolate and use those nanovesicles for some of her experiments. Now, we don't have a Parmesan factory here in Manhattan, but I do have a partner that makes his own yogurt. And we're producing liters and liters of whey that we are bringing up to my lab for Kate and Artemis to develop a method to isolate those nanovesicles and concentrate them into a solution that would be appropriate for making our gels. I'm not going to get into too many of those technical details, but what I do want to share is that we have actually successfully made these materials now, and we're forming these hydrogels using milk extracellular vesicles. And one thing that's really important to note here, and this is something that Artemis worked really hard on, is that the polymer side of the story is also really important. The polymer that used to work for liposomes was not working well for these EV hydrogels, and so Artemis had to re-engineer this. And so what I'm going to show here is rheological data that represents our physical properties of a material. For those of you who are unfamiliar with this kind of data, just look at these blue data points. We want them to be below the dashed line, and that indicates that we've made a gel. So if we look at the polymer by itself, this is a cellulose polymer. We have this hydrophobic chain on it. You can see that by itself it doesn't make a gel. But if we introduce the milk whey nanovesicles and mix them in, it stabilizes the system and it makes a hydrogel. If we remove that hydrophobic component from the cellulose cell, we lose the ability to make the hydrogel. So it's that hydrophobic interaction that's really driving the formation of this material. And what Artemis was able to do is explore different lengths of that hydrophobic side chain and increase its avidity for interacting with these exosomes, and saw that this improved the stiffness of the material and made it behave more and more solid-like, thereby optimizing a lot of its material properties for our applications in drug delivery. The last thing that we ended up doing is looking at how this material would behave in animals. And Artemis started to work with another student in my lab, Ryan, on this. And they wanted to see what this material would look like after a week in the body. And so as a comparison, here's our synthetic hydrogel made of liposomes, 1 week explanted out of a body. And here's the EV hydrogel. And you might immediately notice these red vessels here, indicating that we're seeing some form of angiogenesis. And when we look more closely with histology, we see very clear examples of blood vessels forming. Here in H&E, we see the cells forming these circular structures. In the trichrome stain in blue, we see collagen deposited around the vessel, as you would expect, with the maturation of blood vessels. We looked at this a bit more closely, and it's a little washed out here, so I apologize for that. using immunofluorescence to look at specific markers of angiogenesis. And you can see in this hydrogel, these markers are showing up all over the material. And if we zoom in, we can see some very specific markers, including markers for neovascularization, new blood vessels, and markers for maturing blood vessels. And we see a mix of these showing up in our material. And so with that, I just want to summarize that we have now been able to make these hydrogels from these EV materials. I didn't share it today, but we can do this with other types of these EVs besides milk EVs. And what I really want to highlight is that these hydrogels do seem to retain some kind of bioactivity or biological functionality of their EV building blocks. And with that, I just want to thank the trainees, specifically Artemis and Kate and Ryan and many of others who were working on this. It was really a team effort. And I will cede the floor.
Indeed, please have a seat along with the other speakers up in front here. We have time for some questions until 1:10, I'm told. So, questions?
Hi, thank you so much for your talks. They were incredibly interesting. I'm super interested in biomaterials and especially with like therapeutic platforms. So my question is geared more towards the last two talks. We talked a lot about how we can like manipulate gene expression to control like maybe different phenotypes or differentiation, especially in like dermatology, or control or manipulating antibodies for tailored therapies and multifunctionality. And I was wondering, especially in applications for tissue engineering. I think dermatology is most obvious, but I'm sure it can be applied anywhere where there are a lot of differences between women, men, or also different people of color. And I was wondering how you might think about your approaches to your research to achieve sort of like equitable solutions, or if there's like a one-size-fits-all, how you go about thinking about that.
Excellent question. Thank you for the question. So we actually, I didn't talk about this today, but we developed a machine learning tool called Patches. Please check it out. It's currently on the review in Nature Communications. And what Patches does is it takes some of our transcriptomic data and it predicts what the human equivalent, it can predict what the human equivalent would be. And so that would enable us to actually figure out, because at the end of the day, we're working with animals. And so the demographics that are present in humans are difficult to fully recapitulate in that context. But we're now trying to see if we can actually impute what the human relevance, one-to-one human relevance would be in our data set. It's an excellent question. I think it should definitely inform the way we engineer. In the skin, for example, there are major differences between the sexes. For example, males tend to have a thicker epithelium, which is the top part of the skin than females. The adipose tissue, which is the bottom layer, also is different between the sexes. And that impacts the rate of healing considerably. So, these considerations should be taken into account.
I'll add to that it's challenging to do this super exhaustively in animal models, but in situations where you have, say, organoids or some of the more advanced ex vivo or human-on-the-chip type systems that are starting to evolve and emerge, those are the situations where it's important to consider kind of the ancestral lineage of cells that are used to seed those materials, right? So one of my colleagues, Erica Moore, published a really great paper on the ancestral lineage of cells used in bioengineering that's worth checking out. And this is something you can take a look at so that you can make sure that you're designing experiments that are, as you mentioned, equitable.
One last thing to add to that. I mean, as the first session was talking about, we are developing a quite remarkable biorepository here at Columbia. So we talk about a very wide-reaching base that's going to patch a wide swath of humanity in there. It's an exciting place to be as we start to really start to tackle some of these questions. It's an exciting opportunity. Thank you for that question. Yes.
From our online audience, we have a question from Taylor for Dr. Yusufpour. How long do saRNAs remain in circulation without TMP activation?
Without TMP activation. So the longest that I have done is I have gone to 6 weeks actually, and then I added TMP after 6 weeks, and I was not in all of mice, but in some of the mice, I was still able to get some gene expression back.
I think there's a question back there.
Hello. Dr. Correa, I was curious, because you're getting proteins from like whey from like cows and such, is there any concern about like immune response from the foreign protein, or is that more seen as like a potential benefit due to wanting to create an immune response?
Yeah, it's a great question. It's one that we thought a lot about. And so we were one of the reasons we went into animals is that we want to see, we had used the milk EV system as a model, right? Like, how do we formulate this? I mean, we could do that with other types of EVs that might have more direct and obvious biomedical relevance. But we wanted to see how it would behave in an animal because there's been a lot of interest in using milk EVs as drug delivery vehicles. So, what we saw, basically, we were comparing the hydrogel and also just a bolus dose of the same number of milk EVs just to see if we would see any toxic side effects. We didn't observe any toxic side effects, no loss in weight, nothing like that. The mice seemed quite happy. And then we started noticing in the hydrogel condition this angiogenic response. And so there's a lot still that we need to learn about what we're seeing in this system. And, it's one of those great moments in science where something that you used as kind of maybe like a control or something to help you just get the technology working might actually have some interesting capabilities in and of itself.
Question for Dr. Yosef about your self-amplifying RNA system. How do you construct your RNA polymerase? Is that universal for all your RNAs, or you have to tweak it based on specific sequence?
Are you talking about the synthesizing the original RNA or?
No. When you deliver, you have a replicase.
Yeah,
How do you construct the replicase?
Yeah.
Is that universal for all potential RNA you're using as therapeutics?
So saRNA itself is a synthetic biology platform. So it's derived from virus genome. So we use RNA viruses that have RNA-dependent RNA polymerases. As one that we specifically use is alpha virus. So in addition to our gene of interest, our payload, it also encodes for that RNA polymerase. So once it's injected, that RNA polymerase is translated, and then that's the polymerase that's responsible for amplifying that original strand. Does that answer?
I have a follow-up question.
Yeah.
Can I ask a question?
Yeah.
And how do you deliver the RNA?
Lipid nanoparticles.
Okay, so the polymerase is also RNA encoded?
Yes, the
Okay.
polymerase itself is also encoded from RNA. Yeah.
Thank you for the talk, and just a follow-up question. So, for the in vivo study, I kind of don't remember. Like, do we do the local injection or the systematic injection? And on top of that question, do we still since it's a inducible model by the drug TMP, do we still worry about the leakage and the inducibility in the body?
I'm not sure if I heard the last part of your question correctly. Could you repeat that, please?
Sorry, can you repeat?
The last part of your question. You had two questions there.
Yeah. So, like, since it's an inducible model, right, we have the TMP to induce the self-amplify of the RNA. Mm-hmm. And do we still worry about the leakage and the inducibility when it comes to, like, in vivo study?
Okay, so first question about the injection route. So we are doing intramuscular injection, like how the RNA vaccines are injected in clinic. And for TMP, what I didn't mention is that we cannot force mice to take pills, so we just add the drug to the animal food. And the second question about the leakage you mentioned, right? So that's actually a very great question. So when we design different gene circuits and we screen them in vitro, and a lot of these circuits work beautifully in vitro. But the problem was when we were going in vivo, I was like literally praying that something works and it doesn't leak. The leakage was the biggest issue, and especially because it's a self-amplifying platform, right? So any leakage that you have there can accumulate over time. So that's why we decided that the idea came that we need to amplify is to actually control the RNA copy number too. So that was the additional layer of control that we included there to make sure we get no leakage in the alpha state.
Hi, thank you for your talk. I have a question specifically for Dr. Wopi. So the global skincare industry was valued at over $100 billion last year. And based on your research, what do you think is like the most common misconception regarding consumer skincare in regards to wound healing?
That's a great question. I think there's actually a lot of overlap, and it's happening more and more. So we have a technology that we're in the process of licensing to a beauty company, essentially. And what's happening in Europe is they've banned the use of animals for especially non-essential research, practically every biomedical research enterprise. And so the beauty industry now uses the tools and cell culture models that we use for, scientific discoveries. And so I think that field is becoming a lot more scientifically rigorous, for lack of a better word. And I'm really glad to see that overlap. I think it's the misconception, is it is science, right? It is a tissue, it is an organ, and there's a lot of clinical trials happening that are not being surveyed about RA, the retinoic acid use on skin, what effects does that have? I think there's a lot of data there to be sourced and mined if we apply the right rigorous structures around it. So I think that overlap is something I welcome. Yeah.
Thank you.
We have two questions that have been waiting very patiently on. You gonna read those, or?
Thank you, everyone, for the talks. It was really interesting to hear from all of you. My question is for Dr. Correa. You mentioned that it requires 2 to 10% by weight of nanoparticles to form like these hydrogels. And so my question for you is one, that's a lot of input, and as you mentioned, it can create some issues in terms of exploring the different treatment contexts that you're interested in. And so I'm wondering where does that requirement come from, and has your lab explored any ways to modify the chemistry of these hydrogels to sort of reduce that input amount so you can explore other therapeutics?
Yeah, that's a great question. And this is one of the reasons why in this space you haven't really seen too many extremely over-engineered particle systems, because making a lot of those is challenging. So this is something we're actively looking at in the lab. Like I mentioned, Artemis has been looking at different features of the polymer to see how that can strengthen the system. And so it's something that we're actively looking at. We don't know exactly which parameter is going to be the one that most mediates this, but the reason why this is a need, right, is because the nanoparticle itself cross-links the material, right? And so to have a hydrogel, you need cross-linked polymer, and the amount of cross-linking is directly proportional to how strong that final material is. And so this is one of the reasons why it's one of those things that's kind of difficult to overcome. And so we tend to look at situations where the softer gels tend to be more useful for kind of nearer-term direct translational applications. And it turns out that immunoengineering is actually a great one, because the materials that we currently make are around the same kind of stiffness as lymphoid tissues, right, which is one of the important tissues that you see for educating the immune system.
Okay, now we had two questions online, so please.
So this one is for Dr. Correa also. Are there any concerns with the expression of native surface proteins on the exosomes in the hydrogel, particularly in terms of milk allergies? Are the surface proteins removed, or do they remain in addition to any surface modulations that are added? And then another question, does your research on tissue engineering extend to regeneration of organs like the liver, kidney, etc.?
Yeah, those are great questions. So the first question is can you actually repeat the first question? I'll start with the second question. So the regenerative side of my lab is pretty nascent. We typically have been thinking more about just reprogramming immune tissues as we see them. But this EV material is kind of opening new doors where, maybe we should talk after this. Sure. But one of the things that we're also looking into is if our materials can essentially seed or nucleate the formation of spontaneous lymphoid tissues. And then the first question, can you remind me?
Yeah, sure. Are there any concerns
The
with the
allergies.
expression of native surface proteins on the exosomes in the hydrogel, particularly in terms of milk allergies? Are the surface proteins removed, or do they remain in addition to any surface modulations that are added?
Right. Yeah, so it's a concern. Like I mentioned, the milk system was initially conceived as a way for us to figure out how to make these materials really well, because it takes a lot of materials to do the optimization. Now that it looks like we might have interesting biomedical applications, yeah, the allergy would be probably a limitation in individuals who have milk allergies, because we don't remove anything or strip anything from these materials. But, you know, in the literature on milk EVs, it looks like they actually pass through the gut and can get into different parts of the body, even in tissues like the brain. And so those of you who are consuming milk regularly probably have some milk EVs in your body right now.
We had one question back here.
Hi, I have a question for Dr. Wang. I'm curious, in your system where you're editing the bacteria in the microbiome in situ, and you mentioned that you had managed to modify them so that they were dependent on inulin consumption, and that the population would disappear if you didn't feed it to them. Have you found that, like antibiotic resistance, those bacteria evolve to no longer be dependent on inulin, or is that a potential concern in doing that editing in vivo in the future?
Yeah, that's a great question. You know, we specifically in the context of this, we want to make sure that we link the kind of function of the thing that we deliver to something that we can control externally, and in that context, you know, antibiotics is not involved. And in general, this is one of the challenges in the microbiome modulation field overall, is that, you know, people are using antibiotics as a way to either clear out the microbiota or make large-scale perturbations, which itself is actually quite damaging, right? You're essentially, you know, nuking the environment with the hope that you've like just, you know, altered it in a way that could be useful in a therapeutic context, which generally is not, right? Actually, antibiotics is counterindicated in many cases. In the context of rise of antibiotic resistance with this, we don't think this is necessarily going to be a challenge here, because the payload itself could be transient. It's a transient thing, and so hopefully, you know, we don't have developed resistance genes. But we do show that in a separate study, which we didn't get to talk about, that we can use a similar strategy to actually remove antibiotic resistance kind of genes from native strains. So things like Klebsiella that have carbapenem resistance, which, you know, is widely present in hospitals and things of that, which is a big problem for nosocomial infection. So similar type of strategy is used here.
Okay, unfortunately, we've kind of come full circle from gut to milk back to gut. This is fantastic and quite appropriate because it's now lunchtime. And so in order to keep on time, let's go grab some food. We will be there shortly. More questions. I know there were other hands coming up. Please ask questions over lunch. We will see you there. Thank you.